Hello, welcome to Science Hangout from Science Mission. So we have today Dr. Sandra Rieger with us. She is the assistant professor at the MDI Biological Laboratory. She works on wound healing mechanisms using zebrafish as a model. Recently, her work on chemotherapeutic agent on wound healing and nerve degeneration has gained tremendous interest. She has won several awards to her credit. So in 2003, she won Outstanding Scientific Diploma Research and Thesis Award. In 2004, MBL Scholarship from Marine Biology Laboratories. Again, in the same year, Travel Award from Boehringer Ingelheim Funds. And 2005, she got the citation for Faculty of 1000, which she again got in 2009. She also won Gross Fellowship Award. She also won the Travel Award for the 9th International Zebrafish Conference in Madison. She is also a nominee for UCLA Chancellor Award for Postdoctoral Research. In addition, Dr. Rieger has presented her work in several scientific meetings. She is in the editorial board of several journals and also a reviewer for several journals too, and has published several articles in peer-reviewed journals. Welcome, Dr. Rieger.
Hello.
Hi. So nice to have you on Science Hangout.
Yeah, nice to be here.
So let's have some small chat about yourself, your background, and your research interest. Can you please say something about that?
Yes. So I received my PhD in Germany. I'm originally from Germany. Mm-hmm. And so I went to do my graduate studies at the Helmholtz Center in Munich. Uh-huh. I was in a lab with Dr. Reinhard Köster, who is now at the University of Braunschweig in Germany. And I was studying how cerebellar neurons, granular neurons, migrate from the ventricular zone to other brain regions. And so that's when I first got exposed to the zebrafish as a model, which I'm still working on today. And I noticed that the zebrafish is great for doing in vivo studies, time-lapse imaging in particularly. And so it was very fascinating, this experience to me during my graduate work, that I really always wanted to stay with that model primarily and do in vivo imaging, because it gives so much more information about biological processes compared to looking at static sections in a mouse, for example. And so I really, during my graduate work, I was really looking at the role of cell adhesion molecules and how they guide migrating neurons during their migration to the other brain regions. And then particularly, I was interested in neural or N-caterin, neural cell adhesion molecule, and also NCAM, PSA NCAM in particular. And from there, I went on to do a postdoctoral training with Alvaro Sagasti at the University of California in Los Angeles, where I worked on studying sensory neurons, and in particular how sensory neurons innervate the skin during development, but also later I became interested in studying how these axons regenerate after injury. And so I used the zebrafish tail fin model, because zebrafish have a very high regenerative capacity, so we can amputate the tail and we can study regeneration. And so I was really looking at how axons contribute to regeneration of the tail fin, but also how the injury or the tissue that is injured contributes to axon regeneration. And so I found that hydrogen peroxide was very important in that process. So the skin seems to secrete hydrogen peroxide, and this then leads to axon regeneration. So I'm still working on that particular model today, looking at the mechanisms by which hydrogen peroxide promotes axon regeneration.
And have you tried any other model or just the zebrafish model? So
before I did my grad school, I was also working for about 1.5 years on mice, and then I looked at
Okay.
mouse mutants of human genetic diseases.
Okay. And do you think zebrafish is easy to work on, or like it is just what you thought?
It's definitely cost effective, and also
Okay.
use a lot of animals, because you get about 300 eggs from each spawning, and so we can have very high quantities of when we do our experiments, we can have a high number of animals that we can analyze, which is, I think, very powerful in terms of replication of the results.
Okay. And like when you moved on to the current position, have you thought of getting into industry, or just you thought academics is the better option for you?
Yeah. So when I was an undergraduate, I was actually working in a biotech company in the Bay Area, and I was doing phage display, trying to develop artificial antibodies. And I noticed that industry is very competitive, and the research focus changes a lot. And I thought I want to have a more stable position, at least for a while, to see, to be able to do my own, to pursue my own interests. And I thought academics would be the really good way to do that. That's why I ended up in academics.
Okay, that's a good way to go. And like in addition to research, I know you are busy being assistant professor. You need to work, you need to write grants and publish and all. So if you have some spare time, how do you spend it? What are your hobbies?
Yeah, so I have a 5-year-old son, and I'm trying to spend as much time with him as possible.
Okay.
He's in kindergarten now, but to really look at what he wants and what his needs are. He likes art, so we are doing a lot of art projects.
Okay. That's good. Spending time with the family is more important, I guess.
Yes.
And let's talk about some of the projects you are doing, and you can move on and present your work whenever you get a chance to do that.
So right now I'm currently working on 2 major projects. As I already mentioned before, we're looking at how hydrogen peroxide promotes axon regeneration. Really, the results from those studies, I really wanted to have some kind of an application for that. I mean, it's nice to know what promotes axon regeneration, but how does this help humans? And so I'm very interested in trying to develop treatments for degeneration disorders where axons normally don't regenerate. And so that's how I started to develop zebrafish models for axon degeneration that mimic human diseases. So there's peripheral neuropathy that induces axon degeneration, or it's an axon degeneration disease, which can be caused by many different causes, but we are really interested in chemotherapy-induced peripheral neuropathy because it's easy for us to model in the fish. We simply add chemotherapeutic agents to the water, and we can then study the degeneration processes in the living animal. So that's basically what we are doing right now. But I have also started to develop a model for diabetic peripheral neuropathy, which is also a big problem. So we are trying to develop those models and ultimately test the axon regeneration, the pathways that we have identified there, and test if we can manipulate them in those models to promote axon regeneration or prevent regeneration.
Okay, can you please present your work, please? Thank you.
Yes. Okay, so I would like to start with my first slide, just talking very generally about neuropathy. What is peripheral neuropathy? It really is a combination of, or it can be caused by various different causes, and it can affect various different sensory or various different nerves in the body. One is the sensory system, the motor system, but also the autonomous nervous system. And it typically depends on the cause, on the toxin, for example, or on the disease. It can also cause a variety of different symptoms. And while most of them are associated with nerve degeneration, it also has been found that it can lead to pain formation, but without that nerves degenerate. For example, it can affect pain receptors. So symptoms are typically found in the hands and feet, which can be temperature sensitivity, numbness, acute or chronic pain, and if it affects which are typically effects of the sensory nervous system, but it can also affect the motor neurons, which then leads to weakness, and if it affects the autonomous nervous system, it can then interfere with organ function and ultimately lead to organ failure. Often the respiratory system, for example, is affected, which then leads to respiratory failure. And so what is common to most of those peripheral neuropathies is that the mechanisms are completely unknown of how do these axons degenerate, and how can we promote regeneration or prevent degeneration. And so there's, I just want to have, I just put this list here, which I'm not going to go into detail, but these are just a few examples of processes, of causes of peripheral neuropathy, which are on the top are these chemotherapeutic agents that can cause neuropathy. But then really diabetes is a big initiator of neuropathy. About 50% of diabetics also develop neuropathy over the course of the disease. And there are other initiators such as Charcot-Marie Tooth disease, which is a hereditary neuropathy, for example. So we are really interested in studying how chemotherapeutic agents induce neuropathy for the reasons that I already mentioned before. And so how do chemotherapeutic agents work? There are two different types of chemotherapeutic agents. Some that interfere with replication, and these can be nucleotide analogs such as fluoropyrimidines, for example. And an example is 5-fluorouracil. Those are metabolite inhibitors, which then lead to failure in the replication process of the cell, and this typically leads to apoptosis. And there are these alkylating agents, cisplatin, carboplatin, and oxaliplatin, which interfere with the replication process as well. There are other chemotherapeutic agents that function by a different mechanism, which is they bind to the microtubules in the cell and interfere with daughter cell division. Those are, there are two different types, some that interfere with assembly of the microtubules, such as vinca alkaloids, or for example, vincristine or vinblastine. And there are also microtubule stabilizers, such as the taxanes, and its most prominent member is paclitaxel. And they really interfere with the division of the cells when the microtubules pull apart those two daughter cells. This process is inhibited, also leading ultimately to apoptosis. And so, although very effective in cancer treatment, most of these chemo agents also attack healthy cells because they're just very non-specific. So they in principle attack any cell that is dividing. However, this is also at the same time paradoxic because they attack most prominently the peripheral nervous system, but neurons are terminally differentiated, and so they don't divide anymore. And so the question really becomes, what are the effects in the neurons that lead to nerve degeneration? Mm-hmm. And so there are various chemotherapeutic agents that have been found to affect sensory axons in particular, which are these vinca alkaloids, as I already mentioned, and taxanes. They affect all kinds of different processes in the axon, such as, for example, microtubule, they lead to microtubule aggregation, but also has been found that they can interfere with mitochondrial function, and they ultimately lead to the degeneration of the nerve terminals. And so it has also been found that the CNS doesn't seem to be affected as much, and it is thought that any kind of chemotherapeutic agent doesn't accumulate in the central neurons because of the blood-brain barrier. And so most of these agents, as I already mentioned, they affect the sensory system, and it has been found that the neuropathy starts particularly in the hands and feet, and in particular in the palms and soles of hands and feet. And so we are really interested in the taxane class, or with its prominent member, paclitaxel. And so paclitaxel is used in the treatment of cancers such as breast, ovarian cancer, head and neck cancer, lung cancers, as well as in a variety of different palliative regimens, just to keep the cancer growth in check if metastases have already formed. So it's really used in the majority of cancers that are out there. And it has been found in one study, for example, that up to 97% of the patients were affected by neuropathy. And so, as I already mentioned, paclitaxel affects the sensory nervous system, but it also has been found to affect the motor system if it's administered at very high doses or over a long time period. Sometimes, most of the times, these patients are treated with multiple drugs and over multiple rounds of chemo, and so those accumulated doses and also the timing of the treatment can then also affect the motor system. And what is always the problem when patients are affected, although 97% of the patients in this particular study were affected with symptoms, or they presented with symptoms, not all of them have the same severity. So some of them have very mild symptoms, some of them have very severe symptoms. But those that do have the severe symptoms, they typically have to terminate chemotherapy. And therefore, this also means it's a death sentence for these patients because they just cannot be treated for the cancer anymore. And so it's very important to find treatments which are not available at the moment. And so most of the treatments that are out there are typically to alleviate pain. But there are really no effective treatments that have been found to help or prevent the disease. And so paclitaxel interferes with mitosis. As mentioned before, it really does it by binding to tubulin, to beta tubulin in particular, and these tubulin dimers that form between alpha and beta tubulin, they're normally added to the microtubule plus ends, and it's a very dynamic process. These microtubules typically grow and shrink, and if paclitaxel binds to those dimers and they are added to the plus ends, then microtubules are stabilized and they don't shrink anymore. And this is the process by which they get really rigid and the microtubules just cannot do the function anymore. So they're normally dynamically unstable. They always grow and shrink. But if they are stabilized, then they cannot, for example, pull during the division on the daughter cells to let the daughter cells divide, and then this leads to apoptosis. In the axons, well, this doesn't play a role, obviously, or it has been found, or it's known that axons have a high density of microtubules in them, and what scientists think is that the microtubule stabilization in the axons then leads to microtubule defects in the axons and ultimately to degeneration. So it's known that paclitaxel can lead to microtubule disorganization, for example, aggregation of those microtubules. They are much thicker, the bundles are much thicker. It can also lead to the protein transport machinery changes, as well as binding proteins of the microtubules are altered, and has been found that damaged tubulin subunits are retained. Ultimately, this leads to loss of axonal integrity and axonal transport defects, which then have been thought to cause axon degeneration due to loss of retrograde trophic signals, altered mitochondrial supply, as well as loss of anterograde support processes. It's also been found that paclitaxel can have off-target effects, for example, that are not dependent on microtubule dysfunction, such as it can dysregulate calcium signaling in the cell. But it also has been found to lead to mitochondrial swelling, for example. And there's one study that shows that mRNAs that are normally transported to the mitochondria, that are imported into the mitochondria, the transport is defective in the axons. And this is thought to lead to mitochondrial dysfunction and ultimately to potentially reactive oxygen species. So in cancer cells, it has been found that mitochondria are affected by paclitaxel, and this leads to increased reactive oxygen species. And this could potentially also be the case in axons, leading to axon damage due to increased ROS formation. However, most of these defects really are just observations, and it's really not clear, are these the causes of axon degeneration, even though they are present in the axons, all these abnormalities? Are these really the causes of axon degeneration, or could other mechanisms lead to secondary effects which then lead to those defects? And so we are really trying to analyze this, and I think a very powerful model to do that is to use the zebrafish, because the zebrafish is optically clear, and we can watch axon degeneration in the living animal, and we can analyze over multiple days or doing time-lapse imaging how these axons behave after we treat them with paclitaxel. And hopefully this can give us some more information about the processes that are leading to axon degeneration. And so we have made use of a transgenic line that we have in the lab, which came originally from Chi-Bin Chen's lab at the University of Utah, which is an islet-2B GFP line. Islet-2B is a sensory neuron-specific promoter driving expression of GFP in that transgenic line, and it labels sensory neurons. And you can see in zebrafish, normally humans have two populations of sensory somatosensory neurons that innervate the skin. The trigeminal neurons, which are also present in zebrafish, which are shown here in this cluster, and the dorsal root ganglion neurons, which are shown here in these small little clusters outside the spinal cord. The larval fish have a third population of somatosensory neurons called the Rohan beard neurons, based on the person who first discovered them, which are also present in other amphibians, for example, and also in, they're present in other fish as well as amphibians. And so these Rohan beard neurons are innervating the skin during larval stages, but later the dorsal root ganglion neurons take over the function, which is then very similar to humans. However, in the larval fish, when we do our analysis, we typically look at these Rohan beard neurons. And so they're all unmyelinated, they're sending out unmyelinated axons that innervate the skin, starting as early as 18 hours post-fertilization. And the DRGs are thought to take over the function around 4 weeks of age. And so we use this transgenic line to monitor axon degeneration over time. So we put transgenic larvae in a dish, starting at 2 days post-fertilization. We added paclitaxel to the dish, and then we image the animals every day up to 6 days post-fertilization or 4 days of treatment, and we ask, can we see axon degeneration? And so what we find is that there's a significant decrease in axon branch density in the distal tail fin over time compared to the control animals, suggesting that indeed paclitaxel seems to have an effect on axons, and it seems to be a progressive effect. So we see some degeneration, but it's most significant after 4 days of treatment. Suggesting to us that we can model paclitaxel neurotoxicity in the fish. And what's also very interesting to us is that really the defect is specific to the distal tail fin, which is very similar to humans, where defects are observed in the distal extremities as well. And so we also wanted to know, because of that difference in the sensory neurons that are present in the larvae, can we model neurotoxicity induced by paclitaxel also in adult fish, since in adult fish they are very similarly innervated as humans. And so in this case, we used a treatment scheme in which we injected paclitaxel intraperitoneal on one single injection on 4 consecutive days. And we then looked at, we then stained for axons, because in vivo imaging in adults is a little bit more difficult. So we just did some staining using immunohistochemistry. And we then looked at the effects of axons. So we found that when we inject DMSO as a control vehicle, we see axon branches present in the distal tail fin. However, in the presence of paclitaxel, we see on day 5, 1 day after the last injection, that there's a significant denervation of the fin. And we quantified, again, the axon branch density, or the number of branches in a certain area within the fin. And we find that axons degenerated in the distal tail fin, but we don't see degeneration of the proximal tail fin, suggesting to us that really this effect is highly specific and that it starts in the distalmost region. And we also performed a touch test to assess the sensory response in the fish by tapping the tail fin of these fish and assessing how many times do we have to touch the fish until the fish twitches, for example. And so what we find is that we have to touch over time. We did this during the injection or during the treatment period, that while they started out all very similar, it starts after 2 injections that the fish that were injected with paclitaxel now are less responsive to touch. So we have to touch them many more times until they respond. And we also looked at fish after a 10-day recovery on day 14. And we still find that the touch response hasn't recovered to normal wild type levels by that time, suggesting that it's an ongoing degenerative response, which probably needs some time to recover. So in humans, it's known that the effects are reversible following termination of chemotherapy, but only in about 70% of the patients. So 30% don't seem to recover after treatment. And so we see very similar, that there's more variability by day 14, but still it's a significant decrease. And so so when we first started to use paclitaxel, what we simply did is we used various concentrations of the drug and added to the dish and saw or observed which of the concentrations is most effective in inducing neuropathy. However, we don't know if this is a good comparison to humans, because the metabolism in zebrafish is so much different that we can't really make the conclusion that the concentrations we use is really physiologically relevant in humans. And so, as I already mentioned before, we know that paclitaxel at high concentrations causes motor deficits. So we really wanted to see whether the concentrations that we use, which is 10 micromolar and 22 micromolar of paclitaxel, if these concentrations also cause motor deficits. And we can do that by putting fish into 24-well plates or any kind of multi-well plate. And we have this zebra box from Viewpoint, which allows us to track the movement of these fish over time. And here's just a time-lapse movie showing the tracking information of over 1 hour. And so what this showed us is that when we quantified following different lengths of treatments, that we don't see any significant difference between different treatment regimens in terms of the distance that these animals swam within a 1 hour period, suggesting to us that we don't, that the concentrations that we're using doesn't affect the motor system. Mm-hmm. And so this seems to be very specific to the sensory system, just like in humans, which validates the model to us. So in humans, it has been found that in humans, typically paclitaxel is administered by intravenous injections. There's also nab-paclitaxel, which can be orally administered. But so far, we only have used a treatment in which we added the drug to the water, and it's possible that the skin just sees the highest amounts of the drug, of the chemotherapeutic agent, and maybe this causes all these defects. So we really wanted to have an idea of whether we can deliver the paclitaxel intravenously and then also see axon degeneration. And so we injected paclitaxel at 2 days, 3 days, and 4 days into the fish, and into the cardinal vein, which just runs on top of the yolk. And we then looked at axon degeneration in those animals, and we find that there's also a significant decrease following the third injection with paclitaxel. And we also can do touch assessments, so we simply touch the tail fin of these fish, and then they swim away, and we can assess how many times do we have to touch these fish until they respond to the stimulus, just like an adult fish, and we see that there's a significant decrease in the response, or we have to touch them many more times in order to evoke a response. And this seems to recover following the last injection, and these fish respond normally much faster than in adults, suggesting because the larval fish are still developing, they probably have a much higher regenerative capacity. So one of the interesting observations we had was when we injected the paclitaxel within just a few hours, we found that the tail fins have these morphological abnormalities, and they were very vulnerable to injury. So here's a graph on the right showing that vehicle-injected animals, they have very few injuries, almost non-existent, when we look after a certain time period. And we then also challenged those animals by mechanical stress. So we simply pipetted them a little bit, very gentle pipetting, which we normally do when we mount fish for imaging, for example. What we find is that there's very little injury. However, when we look at either non-mechanically stressed or mechanically stressed animals that were injected with paclitaxel, we now see that the incidence of injuries are significantly increased. And so the question to us was, is there something else going on in these fish when they are injected with paclitaxel? Does this affect potentially the skin? And I want to mention here that the larval or tail fin consists really just of two skin layers, the periderm, which is the outer skin layer, and the basal keratinocyte layer, which is the inner skin layer, the epidermis. And these two skin layers are sandwiched together in the tail fin, and in between there are some mesenchymal cells, and the skin is innervated by the axons. And so there are very few cells here that are present, and to us the question arose whether the skin cells in particular could be affected by paclitaxel, and could this have an effect on axons? And so we also looked at scanning electron microscopy to see what the ultrastructure of the skin looked like in those paclitaxel treated animals. And we found that often we see micro tears in the tail fin of those animals treated with paclitaxel. We sometimes see them in the vehicle control as well, but the incidence of those tears is definitely much more increased in the presence of paclitaxel. And this is just simply after a 3-hour treatment. And when we treated those animals for 96 hours, we find that now the skin cell completely sheds off and exposing actinotrichia, which are in these mesenchymal cell layer beneath the skin, suggesting that the entire basement membrane somehow is damaged as well. And so we think that paclitaxel somehow induces defects in the skin. This is also confirmed by our finding that we see increased stress responses in the skin particularly. So we have this NF-kappa B reporter line, which where NF-kappa B, when activated, binds to binding elements, which then drive the expression of GFP. And we normally see some NF-kappa B upregulation in the lateral line neuromasts, which are it's a sensory organ that is located along the spinal cord. along the ventral region, along the notochord, but also in some dendritic cells. But we don't see normally NF-kappa B upregulation in the skin. However, when we treat with paclitaxel, and I show here with mechanical stress, but also without mechanical stress, we see this increase in NF-kappa B activity in the skin. We also find that that paclitaxel increases reactive oxygen species in the skin. In this case, we used a chemical sensor that is more selective for hydrogen peroxide. And we find that upon mechanical stress, we see these rosettes of ROS formation in the skin. And we think that somehow paclitaxel treatment really induces some kind of a stress response, which could be potentially due to microtubule effects. We also wanted to see how paclitaxel affects the skin in the adult fish, since we also see axon degeneration in the adults. And we wanted to explain that, we wanted to look at the skin in the adults in this case. And so we stained the adult fixed animals with body P ceramide, which is a plasma membrane marker. And we then quantified the length-width ratio, which is a marker of cell shape. And what we find is that the skin cells in the distal caudal fin of adult fish are more elongated in shape. However, when we treat it with paclitaxel after the fourth injection on day 5 again, we see that cells are more rounded, suggesting to us that there are also defects in the skin cells of the adults. So it doesn't seem to be just a novel effect. And so we really wanted to know, could these defects in the skin be explained by an increased accumulation of paclitaxel in the skin, for example? And in order to test this, we actually wanted to see whether paclitaxel accumulates preferentially in the skin, or does it also accumulate in the axon, since it induces axon degeneration. And so we injected tubulin tracker, or also called paclitaxel Oregon green, which is normally non-fluorescent paclitaxel. However, when it gets incorporated into the cell, it then gets cleaved by intracellular esterases, and this releases the fluorescence, and you can detect paclitaxel Oregon green in the cells. And so we injected this into larval zebrafish, and then we used zebrafish that either were injected with a transgene that labels sensory axons in red, as shown in this image here, or into a transgenic line in which keratinocytes were labeled in red. And so we find that paclitaxel preferentially accumulated in the skin cells, but it doesn't seem to overlap with axons. So we see here that there's co-localization with the keratinocytes, but it doesn't co-localize with the axons. And this is, we image those animals up to 12 hours, and after that the fluorescence diminishes, so we can't really test it afterwards. But up to at least 12 hours, we don't see that the paclitaxel accumulates in the axon. So it suggests that it really targets the skin primarily, at least in the early stages. And so this finding suggests to us that, in fact, the epidermis might play a critical role in axon degeneration. But how would it be able to do that? So normally axons that innervate the skin, they are myelinated and then interact. So these axons would interact with the Schwann cells. So how would the skin be able to affect those axons? What is actually the case is that the epidermis in particular is innervated by unmyelinated axons. And these have been shown to directly be in contact with keratinocytes of the epidermis. So a gifted graduate student in the postdoctoral lab where I was in, Alvaro Sagasti's lab at UCLA, did some electron microscopy studies and serial reconstructions of the larval zebrafish skin and the axons. And what she showed here is that these axons are really ensheathed by those keratinocytes. So they wrap around these axons and almost serve the function like Schwann cells do. And it's also been shown in human skin that those epidermal axons are free nerve endings, so they're unmyelinated axons here. And it was in this particular skin section, a skin biopsy, it was shown that Schwann cells are stained here in green with P75, which is a Schwann cell specific marker, and axons are stained with PGP 9.5 or protein gene product 9.5. And you can see that in the dermis, both labels overlap. However, in the epidermis, this is a higher magnification here. You can see that axons are stained in red, but there's no Schwann cell overlap. So this really shows that there's a direct interaction with the axons and the keratinocytes, and perturbations in the skin likely also affect axons. And so studies in rats where paclitaxel was used to look at axon degeneration, they confirm that really the intraepidermal free nerve endings are affected first. So on the left is just a scheme of axons that are myelinated, but once they innervate the skin, they seem to be, or they are unmyelinated or free nerve endings. And in the study on the right, it was found that when paclitaxel is injected into rats in cumulative doses, that the higher the dose, the more intraepidermal nerve fibers seem to degenerate. And so this suggests that indeed our findings might also apply to mammalian models. And so the question is, could epidermal perturbations really underlie cutaneous sensory axon degeneration? And so one proof that we obtained that really the epidermis does play a role is from a pharmacological screen that we started, and I'm going to talk about the experiments that led to the support of our hypothesis that the epidermis is involved. And so we started off with a screen where we looked at compounds that can promote axon regeneration in the presence of paclitaxel. So we already had evidence by then that paclitaxel impairs axon regeneration after injury, and I will show you in the next slide some movies of that. And so in the screen we we used transgenic animals in which sensory axons were mosaically labeled, and we treated those with paclitaxel, and then we added different compounds that we already identified from a previous RNA-seq analysis, which we knew might be involved in axon regeneration. And we then asked, when we amputate the tail fin, can those axons regenerate in the presence of paclitaxel, which they normally can't. And we performed 12-hour confocal time-lapse movies to study axon regeneration. So normally, if we amputate the tail fin in a control animal, you can see how axons within 12 hours regenerate and grow back towards the injury site. In the presence of paclitaxel, axon regeneration is very much blocked. And so we identified one compound, CL82-198, which was able to promote outgrowth of axons in the presence of paclitaxel. And because our screen was a little bit biased already, so we knew what this compound does. It basically blocks the activity of MMP13, a matrix metalloproteinase that is present in the extracellular matrix, and it's a collagenase, so it really degrades. It's known to degrade collagen, and it has also been implicated in other functions, such as, for example, tight junction regulation. And normally, what has been found is in mammals that MMP13 is upregulated in keratinocytes of the epidermis following tissue injury. And it is important there to promote migration of keratinocytes. And so we tested whether we wanted to confirm whether inhibition of MMP13 whether this inhibitor, CL82-198, is really inhibiting MMP13, or is it an off-target effect? And so we tested another inhibitor, DB04760, which is also targeting MMP13 at a very similar site, but has a different chemical structure. We found that both of these inhibitors can promote axon growth over the course of 12 hours compared to paclitaxel alone. And so the next question to us was, can we prevent axon degeneration with these inhibitors? And we co-administered paclitaxel plus either one of these inhibitors, and we asked after 96 hours of treatments how many axon branches are retained in the fin. And so what we find is that with co-administration of these two inhibitors, we can completely rescue axon degeneration. So we can completely prevent degeneration, I would say. And so this to us suggested that if we block the activity of MMP13, potentially MMP13 is upregulated by paclitaxel. And so we looked at quantitative PCR at the gene expression level, whether MMP13 is increased, and we find that with paclitaxel treatment in uninjured animals, we see an increase in the levels, or in the mRNA levels. But in the presence of injury, in combination with paclitaxel, we see this huge increase in MMP13 expression. And we also looked at Western blots to look at protein, because mRNA expression doesn't necessarily mean that protein expression is also present. And so we find that we also see a particular increase in MMP13 expression. And so this is a little bit complicated here, but I just want to walk you through that. What has been, so MMP13 is a metalloproteinase that normally has to be cleaved in order to be active. And the cleaved form has been found in zebrafish to be this 80 kilodalton protein. In humans, there's the uncleaved form. And in humans, this uncleaved proenzyme is 60 kilodaltons, which we don't see. But we do see a faint band at the 80 kilodalton mark. Okay. And we do also see different cleaved variants, and in zebrafish it has been described that there's a 54 kilodalton band, which we see increase, but also a 48 kilodalton band. And we see another band in between here. We don't know. We haven't identified this one. But we definitely see when we take the ratio between the paclitaxel and the control, and we look at uninjured animals, that there's an increase in MMP expression and also after injury, suggesting to us that potentially the paclitaxel, suggesting to us that paclitaxel really increases MMP expression, and that this increased activity of MMP13 potentially leads to axon degeneration. And so we also wanted to look at the cell type specificity or cell type specific expression of MMP13. So when we did immunohistochemistry using the same antibody, we find that there's very low basal activity of MMP13 in an uninjured vehicle control treated animal. But we do see after amputation that there's some increase in the wound margin. And this is very similar to what has been found in mice, where MMP13 is upregulated after injury. However, we do see when we look at these paclitaxel treated animals that now MMP13 becomes much more ubiquitous and much more upregulated compared to the vehicle control. We also wanted to know where exactly MMP13 is upregulated. As I mentioned before, the skin is, mesenchymal cells are between the two skin layers sandwiched in the tail fin. So we wanted to make sure that MMP13 is in the skin and not in the mesenchymal layer. So we used these transgenic, in this case mosaically injected animals, in which we labeled keratinocytes with D as red. And we did a 3D reconstruction, and we find that MMP, which is shown here in green, co-localizes in the layer of the keratinocytes. And we also looked in the paclitaxel treated animals, and we also see that there's more MMP expression and it's also co-localized in the skin layer. We also wanted to know if MMP13 could be upregulated in the axons, because it's still not clear to us whether the effects are really skin-specific or could also axons be affected at the same time. And so we co-localized axons with MMP13 expression using, for the axons, acylated tubulin, which is an axonal marker. And we find that there's no co-localization. So we really think that MMP13 is very selective in the keratinocyte layer, but it's not in the axons themselves, suggesting that potentially really the remodeling of the skin or increased remodeling that is normally not found then somehow affects the axons. And the last slide I want to show is data slide is we looked also in adult fish to see whether the MMP effects are also present in the adult. And this is a little complex here. I'm going to go slowly through it. So when we compare between vehicle and paclitaxel, we basically used a transgenic animal in which basal keratinocytes were labeled, in particular the plasma membrane is labeled with GFP, this P63CAX GFP line. And we stained those animals with the MMP13 antibody to look at MMP13 expression. And we also used Hirsch stain to label the nuclei. And so when we look at the surface of the fish, then we see very little MMP13 expression. We see some red clusters somewhere in between the cells in the vehicle control. And if we go deeper into the tissue, the suprabasal layer, which is the outer skin layer, we see that there are few clusters. If we go into the basal layer, then we see also very few clusters, and this is just a higher magnification. But what we do see, which is consistent with mammalian studies, that MMP13 is present or upregulated in the dermis, which is the inner skin layer. And this is just a higher magnification of the basal epidermis without the basal keratinocyte labeling here. So there's some expression in the basal keratinocyte layer, but it's really high in the dermis. So if we then look in the paclitaxel treated fish, and also this again is 1 day after the last injection with paclitaxel at day 5, we see that now there are more clusters at the surface of the skin, suggesting there's some cell debris. When we look in the bright field images, we see that there's cell debris present. And we see much more of these little clustered dots everywhere. And in the suprabasal layer, we also see more of these cluster dots compared to the vehicle control. And when we look then at the basal keratinocyte layer specifically, which is the innermost layer that is attached to the basement membrane, we see that there's a significant increase in MMP13 expression. This is the higher magnification. This is without the green fluorescence. You can see how much increase there is. Again, the dermis has MMP13 expression, but it doesn't seem to be changed compared between paclitaxel and vehicle control. So it's really, we think that the effect is very specific to the basal keratinocyte layer, and we don't know why at this point. So we also localized or co-localized axons with MMP13 expression, and we see that again we don't see evidence for axonal staining with MMP13. We really just think that MMP13 is upregulated in the basal layer. So the question is, when we co-inject MMP inhibitors into the fish, can we now block axon degeneration in adult fish, which would be the ultimate proof that it's not just a novel effect, that we can potentially also use the treatment in mammals, which is going to be the next step we're going to test. And so we co-administered these inhibitors in the adult fish, and we indeed find that now both inhibitors can prevent axon degeneration significantly compared to the paclitaxel-treated fish. And we also see that the touch response is restored when we compare to the paclitaxel alone treated animals. And so this really gives us hope that potentially these two inhibitors might be effective also in mammals. And so this is the last slide, and where I just want to briefly explain what we think might be happening. So we really think that the effect that we see with paclitaxel is very specific to the basal epidermis. And it's really the basal epidermis that is also innervated by these sensory axons in mammals. And in the uninjured or untreated skin, what normally we see in what we see in fish is that there's some low basal activity of MMP13. And if we then injure the skin, we see our MMP upregulation at the wound margin. And we also know that axons typically degenerate, but then over time they regenerate, or as the skin regenerates as well. So if we look in the paclitaxel-treated animals, what we think is that there's already a higher basal MMP13 expression present, which then leads to degradation of the matrix and potentially also adhesion defects in the skin, which is consistent with these studies in the larvae where we find that they are more vulnerable to injury. And if the skin then becomes injured, this might exacerbate the effect and lead to more increased axon degeneration, ultimately to denervation of the skin. And although MMP13 expression level might go down to this increased basal level that we already found, the axons are impaired in regeneration, so they can't grow back. We also have evidence that I don't show here that this wound healing capacity is also impaired when we treat with paclitaxel. So it seems to be that there's some mutual effect that the keratinocytes are damaged, and this also leads to damage of the axons. And so with this, I would like to thank the people that have participated in the project and also collaborators. We are currently collaborating with Rob Burgess at the Jackson Laboratory, who's doing some mouse experiments to look at these inhibitors in the mouse. And we are also, since the beginning of this year, we have established a collaboration with 2 investigators at the Mayo Clinic, and we are looking at MMP13 expression as well as skin effects in human patients treated with paclitaxel. And with this, I would like to finish the presentation.
Okay, thank you so much.
Hi.
So it's a nice presentation.
Thank you.
I have a couple of questions.
Mm-hmm.
One is you had done this treatment both in vivo, that is injecting into the vein, as well as in vitro, like in incubating.
Mm-hmm.
So do you see the similar effect?
Yes.
313?
So ideally for us, the best way for doing in vivo studies is to just simply add it to the water. And so that's why we wanted to compare these different systems so that we can make sure that we see the same in all of the different methods. And if we do, we can use that incubation model ultimately to do the more faster experiments.
Yeah, why I'm asking is just when you add this thing to outside, it might be due to some other effect.
Yeah, we
so
know,
if you add
it can be taken up into the mouth already. They have developed a gut system, at least by 4 days or 3 days, they can take it up.
Okay. Do you know anything about the metabolism of this toxin in fish? Fish? No?
We don't know.
Not much.
I mean, the only thing we can say is that when we inject the paclitaxel Oregon green, the tubulin tracker, that the fluorescence goes away within 12 hours. But it doesn't mean that the drug gets metabolized. We don't know if it's simply the effect on the fluorescence or is it really the metabolism that eliminates it. But it could be potentially 12 hours.
Yeah, okay. Because in humans, like liver metabolizes all these into new metabolites that may be toxic.
Mm-hmm.
So all those things, okay.
Anything.
Yeah, this is a much simpler model.
Yeah.
And any other metalloproteinases are affected, or it is only specific for MPP13?
We haven't tested. So it's a little bit difficult to test other metalloproteinases if we don't have a mutant, for example. So inhibitors, they're very non-selective, most of them. So originally, MMP inhibitors have been created because MMPs are upregulated in cancer, and the active site, the zinc binding site, is very similar between all of them. So people that use inhibitors for one, they usually target a lot of other ones as well. And so there was a second generation of inhibitors that has been produced, especially for MMP13, which targets not the active site, but an S1 pocket, which is right next to the active site. And it has been found that this doesn't completely eliminate the activity, but reduces it to a great amount. And I think in our case, that's the key, because we think that, because we know basal activity of MMP13 is present, and if we would completely block it, it might just give us a completely different effect. And so I think we just bring it simply back down to its normal activity, which is critical in this case.
That makes sense.
And so we can't test other MMP inhibitors so far because they're not very selective. And so these are the two that are highly selective.
Okay, I think I don't have any more questions. And thank you very much again for your time and effort, and I really appreciate it. And if you have more results in the coming, free, so we can have another one, so that people are getting much more from your work in your lab.
So right now we're actually looking at the effects of paclitaxel on microtubules in the keratinocytes to see. Potentially, I think that the microtubules definitely play a role, but it might just be that the effect is in the keratinocytes and not in the axons. And so we have evidence for that, that reactive oxygen species might play a role in MMP13 expression, because we know from previous studies, RNA-seq studies that we have performed, that hydrogen peroxide can promote MMP13 expression. So we think there might be a link. So that's what we're looking at, and hopefully another publications coming out soon.
That's good. And it looks like since the keratinocytes are damaged, axons cannot denervate into the
that's what
skin. That's what is happening. It's not the nerve per se, it is the skin which is affected.
I think it might be a dose dependence, that at very low concentrations that we see these effects, but if you treat with high concentrations, you will get damage also in the axons themselves, and not one specific. No, it's a dose.
And you mentioned that you'd be working with the patients or something like that to get some patient samples.
Yes. So hopefully this leads to some interesting results.
Yeah, that would be interesting if you can see the similar effect in humans also. That would be very interesting.
Yes.
So again, thank you so much, and we look forward to more of your work, and good luck with your all the projects.
Thank you.
Talk to you soon then.
Bye.